Hello, folks. Welcome to the lecture on macromolecules. What I'm going to do today is use this screencast format to sort of introduce you to these macromolecules in a light sort of way, I guess, and look at their structure and their function. These are the four major, they're called molecule types. Like these are just big molecules because they involve a lot of different atoms in them. But each of them share some properties. So we're going to talk about that today and what function they provide. I'm going to begin here at the top with carbohydrates, which are essentially sugar polymers. A sugar you may know as C6H12O6 looks like this little guy here. This is called a hexose sugar. It's represented in a ring structure. I like to think that each of these molecules has a picture that goes with it. You can draw like a cartoon or an image, and this is the image of a sugar. And if you take one of those sugars and you link them together like this, 1, 2, 3, 4, 5, 6, and then 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6. This is a polymer. All right, and carbohydrates, of course, store energy, right? This is energy storage. And we find them in foods, and the way we find them in foods is often as either starch, primarily, which we find in things like potatoes. Depends on, we have pasta, we have bread. And the issue is that essentially these sugars were being stored by these plants for use for growth and reproduction. And when we harvested their storage structures, then we can eat them. And so we get to eat the carbohydrates, the stored starch of plants. If you look at the way these molecules are connected, I'm going to go down and give you a couple of ideas about carbohydrates. Let's go down here. I wonder if I can take this little drawing here and copy it. How about that? I love using my fancy tools here. So let's try if I do this, and then I just, maybe not that easy. Copy. Well, maybe not that easy for me to do. That's okay. In any case, I can redraw it very quickly and very easily, which is what I like doing anyway. So let's go down here to sugars. And 1, 2, 3, 4, 5, 6. And so this is my hexose ring. And then when I put it in a series, you know that sugars, like this is an example of glucose, which is a hexose sugar. All right. And if you put them in a series like I just did a moment ago, 1, 2, 3, 4, this series then becomes something like spaghetti. I know. So spaghetti is essentially long, like thousands of glucose molecules long. But when you taste spaghetti, it doesn't taste like sugar. Now, that's because in your mouth you have an enzyme called amylase. And it can make spaghetti taste like sugar because this is a starch, right? And what amylase does is it grabs on here like a great big toothy thing. And it breaks these things in half and cracks them. So it frees them up so they then become, once again, single glucose. Oops, I wanted to make them single glucose pieces, so I will do that. All right, so this one, and then it makes, this one's by itself. Right, and now they are glucoses again, all on their own. And these glucoses are sweet. So if you take a soda cracker, which is a bunch of starch, and you hold it in your mouth for about 2 minutes, you chew it up and let the mush sit in there. The mush gets sweet. You can do this taste with bread, you can do it with pasta, whatever you like. But it's a good example of how enzymes, amylase, which by the way is a protein, enzyme, which does the job of breaking things down in this case, and then it makes it sweet. So that is how you can remember that carbohydrates are sugars, because pasta is sweet. Now we're going to move on to lipids. Lipids are the next group here in our group. And the symbol for the lipid that I'm going to give you two symbols for lipids. My favorite symbol is one that I call the international symbol for the phospholipid, which looks like that. And I can put a circle around it. And that's my phospholipid. Right? Another symbol might be something like this, where you have something like that, which is a triglyceride. All right, and in a few minutes, I will show you some examples from the PowerPoint where you can look at their actual drawings of these things. For now, let's page down and take a look at lipids. And I'm going to talk about them as they are a glycerol head. I should tell you there are many kinds, right? Some lipids, which I won't talk much about, are steroids. And these are communication molecules, which when they float through the body to other areas, they have a physiological response. Those have a lot of unique shapes, which I won't talk about, not today. And the other ones, though, are fats, and that's what I'm going to talk about today. So that includes triglycerides and phospholipids. So glycerol head is on the top, and then it has what they call a fatty acid tail, or tails. So what that looks like when I draw the circle is the glycerol head is actually charged, right? So it's a polar, or at least it's polar, which means it has a positive side and a negative side. And then it's attached to, at the bottom, we draw them like these square structures like I showed you there. But these square structures are actually long chains of hydrocarbons, which means you have a chain of carbons down the middle like this, C, 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 and they're all linked with covalent bonds. And then off to the side of each of those carbons is a hydrogen, a hydrogen, a hydrogen. And again, my drawing of these molecules is going to be a little bit rougher than a picture that you could see online, so you could look that up now or wait. So each of these is the same, right? You have this chain of carbons down here with all these hydrogens on the side, linked like this. So, you know, we call these hydrocarbons. There's a lot of energy stored in fats, and yeah, they're kind of similar to gasoline, actually, and hydrocarbon fuels. But the important part about them is that this tail is non-polar. So what that means is that if you were to put a molecule like this into water, what would the water do? Well, water molecules, remember, are polar, so they're going to fly over here and they're going to love this end, and they're all going to go around here and have a good time and stick close, right? But at this end down here, water is not interested. Because water is polar, it doesn't have any attraction or unattraction for all of these hydrocarbon tails. So what happens when you drop a hunk of fats into water is that they end up being organized such that all the heads are on the outside like this, but they have their positive, negative, they have their positive, negative, and then they have on the inside, they have all of their fatty acid tails. I guess I give you this because it provides you with some idea of how in nature we might have some self-organizing structures. So it makes a blob like this, and all this has no water in the middle. So if I enlarge it, no water in here, but there's lots of water out here. So you see lots of H2O floating around and getting friendly with all these things we call hydrophilic heads. Now hydrophilic means water loving, right? And the hydrophobic tails, essentially what we talked about, cohesion and adhesion, the tails are, the water is so drawn to itself that it basically pushes itself together, forcing the tails away, largely because they're just not interested, right? And so these tails all get pushed into this thing, and this makes a little round blob, right? And what's kind of cool about the blob is in nature they call this a micelle. They are self-organizing, right, which means a little cell, and it is essentially a blob that has an order to it as a result of entering into water. So this is a way that we can bring the structure of water together with the structure of fats, and we can look to see how they cause these fat blobs to occur in water. So if you put fat molecules into water, they end up together because they're being excluded by the water, and they end up in these little balls. All right, and now I'm going to talk a little bit about another really important type of fat, which is the, I guess I can't skip ahead. I'm going to go to my phospholipid in a minute. I want to return to this guy. In my lecture in a moment, when I talk about the PowerPoint, we'll talk about this case where I have a, let's enlarge one of these and take a look over here, okay? Carbon, 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 hydrogen on all sides, right? So when it makes this sort of structure, this fatty acid tail, the fatty acid tail is straight. You notice how I drew this straight. What can happen, and this is called a saturated fat. Saturated with hydrogens. So they're all there. If you remove a hydrogen, it will change the shape. So let's take this hydrogen off, okay? And then what happens is the chain will change shape so that it goes like this. It'll get to this carbon, and the carbon will change shape right there. There'll be an angle. So we're going to miss a hydrogen, and what will happen is this carbon will make a double bond here, and there won't be a hydrogen where there was supposed to be one there. And then we'll have one over here where they were before. Now this is kind of a weird thing to have happen. So you have a hydrogen out here, but not, there's the missing one there, right? And now you've got them here. Now what this causes is a bend in the chain. So now it's an unsaturated, this is an unsaturated fat because unsaturated fat. Now you can read these on your food ingredients, and that's essentially what it means. The structure is different, it's curved. And what that really means in the long run is that if you have a bunch of them that are shaped like this, they stack together really well. So the saturated fats are solid at room temperature, right? But if they have a crooked leg like this, they don't stack very well, and so they make liquid at room temperature. So this is the difference between saturated and unsaturated fats. Saturated fats also tend to cause heart disease, and unsaturated fats not so much, probably partly because of the way they stack. You can think of these guys as stacking like a box of pencils, and these guys like a box of worms. Worms and pencils, all right? But the worms are soft and move around, and the pencils, well, they can cause a blockage. So that's a really nice structure-function relationship between the hydrocarbon tail and the hydrogens and the actual solid or liquid form that we see at room temperature. The last topic I'm going to go on to is my favorite phospholipid topic. And so it looks like today I'm only going to get through the first two of these macromolecules, but let's talk about phospholipids. because I just really like them. Okay, woo! I make this little bent leg because there's an unsaturated fat there, positive and a negative. Now, I've already mentioned the Micelle formation, and this is a particular type of fat. It's glycerol head on the top, which is polar, and the nonpolar fatty acid tail. Okay, there's a pair of them in this case, and this forms one of the most common structures in eukaryotes, which is the phospholipid bilayer. Phospholipid bilayer, which equals the cell membrane. That's the cell membrane. How does that work? Well, it works because, and it stays in place because it is a series of the glycerol heads on top, and then their bodies underneath, like this, their tails. Boy, there's a lot of those. Almost done. Okay, I'm getting kind of good at drawing those. And then notice their charge, plus, minus, plus, minus, plus, minus, plus, minus, plus, minus. And these line up for some interesting reasons. Anyway, and then we go down here, and on the other side is another row of phospholipids like this that line up, and they also have a fatty acid. I'm going to get faster at drawing my fatty acid tails. Okay, so they also have fatty acid tails on them. And so this makes the plus minus, plus minus, plus minus, plus minus, etc. This is the bilayer, and you can see that we get water on this side, and we get water on this side, but no water in the middle. And so this little sort of two-layer pancake design makes the cell membrane. And you can see when it's in water, with water on the inside, water on the outside, this fatty acids in the middle here become the super important barrier between the inside and the outside of the cell. Now keep in mind that you can put holes in this. You can't cross that, but you could make a protein hole like this and then keep going on the other side, right? So you could keep your membrane going like this. And we'll talk about this later. This becomes an integral membrane. And this could like send little ions through there like this. Woo! Could go through that hole into or out of the cell. So that's the cell membrane. And you can imagine it wrapping around a cell. Whoop! You know, it made this membrane like we just saw here. Okay, and this is the inside. And this is the outside. Right? That's pretty awesome. That's my cell membrane. All right, so that is 2 of the macromolecules. I've got to go to another class right now. Thanks for tuning in.